Hello, everyone. My name is Dr. Lena Sirik. I am an associate professor in environmental engineering at University College London. I'm a microbiologist by training, a microbiologist, but I actually work in the Department of Civil, Environmental and Geomatic Engineering, and I study how microbes interact with the spaces within which humans spend their time. So our offices, our schools, our homes, and our hospitals. So before I get going, I want to thank ProEconomy for inviting me to do one of these webinars. Thank you to Byron and Sonia. I'm delighted to be able to tell you about some of our work, and I'm going to talk about what role surfaces might play in the spread of pathogens. And this work that I'm going to present to you is mostly within the hospital environment. In fact, it's exclusively within the hospital environment. I guess thinking about how microbes or where microbes might lurk within the built environment has become extremely topical within the past few months, and everyone now, is aware that you might pick something up from a surface or it might be in the air. But really, this was only really somewhat investigated in hospitals, and even there, not to the extent that you might expect. So there's a few different little bits that I'm going to talk to you about. There's sort of a few small separate studies, but they're all connected to each other, as you'll see. So first of all, I'm going to talk a bit about what we found when we carried out some microbiological sampling of surfaces before and after cleaning in a pediatric outpatient ward. I'll tell you a little bit about a cleaning audit that we did where we observed some cleaning practice, then carried out some training and then observed practice again. Talk a little bit about what role the material itself might play in the spread of pathogens. And then finally, I'll talk about a study that we carried out in the same outpatient pediatric ward as the first one that I'll show you, which is looking at what basically how contamination, and this was in the form of a piece of DNA, spread throughout the ward over the period of a week. Okay, so I shall get going. So first, surfaces before and after cleaning. So this was a study that we carried out in an outpatient ward. So the patients come in the morning or at some point in the day, stay for the day, but then they go home at the end of the day. So nobody is staying in overnight. And what we did was take samples from the surfaces, and these are all of the different surfaces. You can see them on the map. So on this map, you can see the layout of the ward, and then you can see all of the different surfaces that we sampled in each of the different spaces. So for example, if we look on the top left, we've got the toilet there, so sampled the window ledge, the door handle and the bin, and then there's the sluice, so there we sampled the sink area, the door handle and the windowsill, and so on. I won't go through all of them. So what we did was take these samples in the evening at about 6 o'clock when all patients had gone. Then the cleaning of the ward took place, and then another set of samples from exactly the same places was taken after the cleaning was finished. And we used contact plates for this. So these are sort of normal agar plates, but they have a slightly convex surface. So it means they sort of sticks out beyond the plastic, and just press these onto a surface, hold them down for about 10 seconds and then lift them up. And we just looked at total viable count. So we didn't look to identify any of the microbes or anything like that. We were just looking at numbers. Okay, so here are our results. So these results, these first set of graphs is looking at the numbers of colony forming units on each plate before cleaning. So this is just showing you the levels of contamination on the different types of samples that we took. And I've split these according to different criteria. So we've got the area, and by that I mean, is it a clinical space? Is it a bed space? Is it a non-clinical space like the reception area? Is it a sink? So is it a place that is often wet and humid? Then also we look at material. Who are the people that touch these surfaces the most? So is it the patients and their parents? Is it staff? Is it everyone? Or is it a place like a windowsill that really nobody touches much? Then we also looked at whether there's an impact of who cleans. So who is responsible for cleaning that particular area? Then risk perception. So is that surface thought to be high risk and therefore people are more conscious that they should be cleaning it? And then finally, cleaning difficulty. So is the surface perceived as being difficult to clean and therefore maybe not cleaned as well or clean better because the cleaner is conscious of the fact that it's a tricky one? So then looking at the actual results, so if we look at the top left first, so looking at area, so the highest counts of bacterial cells that we found were in the non-clinical areas. So these were areas like the reception area and play areas. But there was still quite a lot of variability amongst most of our samples. We also found not, I wouldn't say a high, but we found a higher level of contamination in the sink areas, which makes sense as these are wet and humid and a good place for bacteria to thrive. And then from the point of view of clinical spaces and bed spaces and non-bed spaces within areas where patients were of a similar sort of level of contamination. And then looking at material, the most contaminated materials were vinyl materials, and these were generally the floors, so that makes sense. And then after that, we had ceramic and coated wood. So the ceramic is generally going to be sink area, so that goes along with our previous results for sink contamination. And then coated wood, I think these areas were mostly in the reception area or tables that the children might play on. So again, these were fairly highly contaminated. And then again, you can see, just as I was saying there, depending on who touches. So we found that the surfaces that were the most contaminated were those that were touched by patients and their parents. And then the ones that were least contaminated were those that were touched exclusively by staff. And again, that makes sense because we would expect staff to be adhering to good hygiene, hand hygiene. So we would expect those surfaces to be the least contaminated. And then looking at who is responsible for cleaning. So in this case, basically the majority of the surfaces within the ward are the responsibility of the cleaner. After that, we have the surfaces that are cleaned by clinical staff. So these might be things like weighing scales or changing tables within the height and weight room. They are also medical devices that the clinical staff will clean. And then play staff are responsible for cleaning only the toys and play areas. So you can see the areas that are the responsibility of the cleaners were the most contaminated. But ultimately, these are areas like the areas in reception, the floors, and so on, which are thought to be of a lower risk. And then thinking about risk perception, so spaces like high touch surfaces are generally thought to be high risk, and these were found to be the least contaminated, and surfaces that were thought to be low risk were the most contaminated, and that makes perfect sense in the context of this study. And then finally, looking at cleaning difficulty or the perceived cleaning difficulty, the surfaces that were the most difficult to clean were the cleanest, and then those that are moderately difficult or easy to clean were more contaminated. So all of this is published in a paper, and you can have a look at that in more detail if you like, if you follow the reference below. So another way to look at the data. So what we were looking at here actually was how good the cleaning of the surfaces was. So this graph is a little bit tricky to understand. So what we have here, so on the x-axis on the bottom, we have colony forming units before cleaning. So how dirty was it originally? So to the left, we have the cleanest surfaces originally, and to the right, we have the dirtiest. And then on the y-axis, on the left, we've got the percentage change in colony forming units after cleaning. So the more we go into the plus percentages, the dirtier the surface got, and the more we go into the minus percentages, the cleaner the surface got. So basically what this graph shows us is that for surfaces that were very clean at the beginning, the majority of these got dirtier with cleaning, whereas for surfaces that were moderately dirty to quite dirty, these got cleaner. So what it basically tells us is that cleaning is almost like more like a smearing to a good level rather than getting everything very clean. So, you know, that was somewhat concerning. So we also carried out some work to look at how well the cleaning is performed. And now this was done in a different ward, in a high dependency ward, so the context is different. However, the theory is pretty much the same. So what we did in this case was first to carry out 50 hours of observation of cleaning practice before any training was done. And then a 15-minute training session was given to the various staff that carry out cleaning. And then another 50 hours of observation was carried out to see whether the training had made any difference. So what we were assessing the cleaning on was this 5-component cleaning bundle. So the first component was, has the entire surface been cleaned? So that's making sure that the whole surface is cleaned rather than just one particular area. Then was an S-shaped motion with overlap used? So this is to make sure that the entire surface is cleaned and that no areas are left dirty. So it's a motion that's used to make sure that everything is cleaned properly. Then was one wipe used for each surface or until dirty or soiled? So this is making sure that you're not using the same wipe on multiple surfaces and potentially spreading contamination from one surface to another, and also to make sure that if a surface requires more than one wipe to be used, that they're used. Another component of the bundle was looking at whether the wipe was entirely folded out. So the wipes are designed so that they to work best if they're totally folded out, and this means that you have the best what's the word you get the best amount of contact between the antimicrobial within the wipe and the surface itself, and also that you have the best chance of transfer of the antimicrobial onto the surface. So they need to be used as they were designed to be used. And then finally, was the correct wipe used? So depending on what surface is being cleaned, the right sort of wipe should be used, because it might have antimicrobials within it that you don't want to introduce onto a person, and so on. So some of the cleaning was just usual surface cleaning with a mop, or, you know, cleaning things like tables around the patients or the bed rails. But sometimes these were medical devices which needed to be cleaned with ethanol and aren't suitable for being cleaned with the usual sort of surface wipe. Okay, so that was our 5-component bundle. So this is what the cleaning was assessed on. And then the training itself was based on this 5-component bundle. And again, the cleaning after training was assessed on these 5 components as well. So our results. So these show you the score of the 5 out of 5 for the 5-component bundle. So if for all of the 5 components, they would get a point if they hit any of them. So you can see in the black color, or is it dark blue? I can't see. So this was the score before. So the score before was somewhere just over 2. And then after the training, the score moved up to an average of about 3. And this was for all of the staff that we worked with. And the main staff that we worked with were cleaners and also nurses. So within this ward, I think there are about 5 or 6 cleaners who always carry out the cleaning in this ward. There are probably about, I think, over 100 nurses who work on this ward, so depending on what shift pattern they're working. So we managed to train all of the cleaners, and I think about 80% of the nurses, or I think it could be a little bit, and clearly there was an improvement after the training, not a huge improvement, and still most people weren't getting a score of 5, but it was certainly an improvement, which is important. Especially since this was only a 15-minute training session, so pretty short. So here we have the results looking at the cleaners and the nurses separately, and looking at which bits that they did well on and which bits they didn't do so well on. So on the whole, even before training, wipes were folded out pretty well and the correct wipe was generally used. The entire surface wasn't necessarily cleaned before, but after this improved. They did pretty badly on using one wipe per surface and the S motion. I mean, both of these improved after training, but was still pretty bad. So just over 20% after training. So still not ideal. And then for the nurses, for which the data you can see on the bottom graph here. So again, pretty good. Oh, I'm afraid I've cut off the before and after. So the after is the lighter blue and the before is the darker blue. So an improvement certainly in the correct wipe being used, although this was pretty good at the beginning also. And using one wipe per surface was also pretty good before training, and a significant improvement was seen after training. Then for what the wipe being completely folded out. So this was about 50% adherence before the training, actually dropped a bit after training, possibly because they were concentrating on other things like using one wipe or using the correct wipe or making sure they do the S-shaped motion and getting the entire surface cleaned. So that was interesting that one got worse after training. And then you can see the S motion and cleaning the entire surface improved quite dramatically within the nurses after training. Still not ideal, but better. Okay, so now a bit about surface materials. So I'm not going to present our own work here, but I just wanted to talk a little bit about surfaces and what characteristics they might have that make them easier to clean or more difficult to clean or more susceptible to having microbes lurking on them. So the first thing to mention is surfaces at the microscopic level. So on the right, this is a piece of stainless steel, and we think of this as being a very smooth, non-porous surface. But actually, if you see the little scale bar at the bottom, the little white bar, that's a 10 micron micrometer size. So you can see that actually at the micron level, which is the level of microorganisms or bacteria, certainly, there are quite a few grooves and nooks and crannies for these microbes to wriggle into. So we do need to be careful about how we clean these surfaces. So from a point of view of cleanability, a rough surface is certainly more difficult to clean than a smooth surface, and a porous surface is certainly more difficult to clean than a non-porous surface. So ideally, we want to have smooth, non-porous surfaces that we can clean easily, and we want these to be durable and resilient to cleaning and the chemicals that might be contained within cleaning products. We need to be quite conscious of high touch surfaces, so those that are touched by a lot of people consistently, and these are generally thought of as high risk and are cleaned and might even have a more comprehensive cleaning regime, a more intensive cleaning regime. Something else I want to talk about is antimicrobial surfaces, and I think there's a lot of this, especially at the moment during COVID, that everyone is selling antimicrobial this and antimicrobial that apparently kills 99.99999% of everything, and will make sure that you're safe for, I don't know, I mean, some claims say even up to 30 days or even years. So some of these products certainly do work, but the majority of them is tested according to standards, which are performed in laboratories and which are performed with a number of standard pathogens, which are well studied. So it's really difficult. So although these might work very well in suspension, where you basically mix up your antimicrobial with the bacteria and you take some samples and 10 minutes later everything's dead, that's quite different to how that antimicrobial might interact with bacteria on a surface as opposed to in a liquid where there's a lot of contact, and also on a clean surface. So some of these standard methods are performed on surfaces, but these surfaces are totally clean, and these studies are performed under controlled conditions. Surfaces in the real world can be quite different. They can have dried biofilms, they can have other soiling there that can stop the interaction between the antimicrobial and the microbes. So there have been a few studies which have looked to see whether using antimicrobial surfaces within healthcare settings has an impact on the incidence of healthcare associated infections. And most of these have found that the studies that they've reviewed have been done in a substandard way. So it could be that the sample set is too low, so you can't really draw any significant conclusions. It could be that it was not done in a controlled way. There was a lack of blinding, so the researchers were expecting to see a certain result, and therefore could have skewed the results to some extent. So often different methods were used in different studies, which you then can't compare. There was certainly lack of data in long-term efficacy, so often this was done on a very short-term basis, a week, a few days, no more than a month. And there's also a lack of data on how durable these surfaces are and how well they withstand being cleaned day in, day out by various chemical agents. Okay, and finally, I'm going to present to you some work that we did quite recently. So this was just before the first lockdown occurred in March. And what we wanted to do here was track how contamination on one single surface might spread within an outpatient ward over a period of time. So what we did in this case was to design a DNA oligonucleotide. So this is basically just a chunk of DNA, and it was based on the sequence of cauliflower mosaic virus, which we thought would be totally safe to use, and has in the past been used within the healthcare setting as a sort of surrogate for contamination. So this was just a linear DNA at a copy number similar, well, about 10 to the 8, 10 to the 9 per milliliter. So we basically spotted a bit of this onto a bed rail in an isolation room on a Monday morning. And then we sampled 44 surfaces around that ward, not the surface that we inoculated originally, because we didn't want to remove the oligonucleotide from there. So then we sampled these 44 surfaces around the ward each evening during that week. So Monday evening, Tuesday, Wednesday, Thursday, Friday, and then we stopped. And something that we'd done before this release was to check that the oligonucleotide, this marker, was easily removed with normal cleaning practice, so that the wipes, the antimicrobial wipes, totally remove it, and also hand washing totally removes it, using hand sanitizer totally removes it as well. So we wanted to just to make sure that it wouldn't withstand the normal cleaning and hand hygiene practices, and it didn't. Okay, so here are our results. So if we look at the bars first, no, take it back. If we look at the overall positive first, so that's the green line, and that corresponds to the y-axis on the right, so the overall positive sites in percentages. So on day 1, so this was 10 hours after original inoculation onto that bed rail, about just over 40%, I think about 42% of the 44 surfaces that we sampled within the ward were contaminated with the oligonucleotide. On day 2, this fell a little bit to just under 40%. On day 3, it rose to 52%. On day 4, it fell again down to somewhere between 30 and 40%, and we ended on 42% again on day 5. So clearly there was some inoculation and then removal and re-inoculation of different surfaces. We can't say anything about how the movement of these oligonucleotides happened, because we have no idea. All we know is that on day 1, they weren't there, but then on day later on that day, they were in all of these places, and then on day 2, 3, 4, and 5 as well. And we also resolved the data a little bit. So this is looking at the bars now, and that's corresponding to the y-axis on the right. Sorry, on the left, which is the positive sites by area. So here we were looking at the immediate bed space. So this would be the bed rail and the table over the bed rail. Then the wider bed space, which might be the door handle within a particular cubicle, or the door handle in the toilet in there. Then clinical areas, so these would be the treatment rooms where the treatment is administered to the patients, and then general wards, so this would be the reception areas and play areas and so on. So you can see that the immediate bed space was very often quite contaminated, so consistently contaminated with percentages somewhere in the 50s throughout the week. And also at the beginning of the week, well, on days 1, 2, and 3, you can see the clinical areas are particularly contaminated, especially on day 3, where 80% or over 80% of those were contaminated with our oligonucleotide. So that's basically the gist of this data. We're still working on this at the moment, because we actually inoculated 3 different oligonucleotides to 3 different places within the ward, and we need to now unpick where each of the 3 went over this time on all of these 5 days. And it's quite difficult to do, because we have a number of variables. So we have both spatial and temporal changes. So we are working on it, and hopefully soon we'll have some animations done of this, which we'll publicize so that everyone understands how these things move around. But the crux is that the oligos spread very quickly and consistently, and clearly cleaning regimens and hand hygiene were not necessarily as good as they could have been. So to conclude, surface contamination and cleaning is a really complex issue. There are many different factors at play here. And in order to tackle it, we need to use a holistic approach. So we have things like design, how we design our wards, and how we design them to be used. So how do we design them both from a physical point of view, but also from the point of view of what they need to, the function that they need to perform, and what they need to provide for the patients and the healthcare staff. But it's also really important to educate both staff and patients, so they understand why they have to do the things that they are asked to do, because I think if they understand why, then it's much clearer to them that it's important, and it's much more likely that they will remember to wash their hands or clean a surface and so on. And then finally, collaboration. I think it's difficult when there are lots of different people working on a specific issue. So there'll be architects, there'll be engineers, there'll be building contractors, there'll be clinical staff, there'll be estate staff, there'll be infection control staff, there'll be patients and so on, academic staff like myself, and we all speak a slightly different language, and it's sometimes difficult to understand each other. So it's really important that we work together and try to understand each other better to have a better result for our patients. And then finally, I need to thank everyone who worked on all of this work. So the first person to thank is Stacey Rawlinson, who you can see in the top left here. She basically carried out most of this work. And I need to thank Melissa Canales, who is our research technician and helped with the work as well. And then the wonderful Elaine Cloutman-Green, who you can see on the bottom left, who is Stacey's other PhD supervisor. And then Helen Siraki, Faiza Sadi, Barbara Childs, and John Hartley at Great Ormond Street Hospital, who worked with us on all of these studies. And thank you all for listening.